Hello, Marcus Leach here, and today for my digital storytelling project, I will be telling I guess you could say, story to help you better understand the process of turning DNA into proteins through the central dogma of biology. Okay, so once upon a time, there was a blue penguin, and this blue penguin had a trait of being blue. And this was determined by a gene that the penguin got from its parents. And so basically the way that that gene was transcribed from the DNA in the penguin was, first, along the DNA there was a spot called the promoter, and this is in the step of initialization. And what the promoter does is it is a signal, a place where the RNA polymerase is able to hook on to the DNA strand and then start the process of transcription. The next step is elongation, and basically this is the part where the RNA polymerase is actually copying the DNA and forming what's called codons. And codons are a group of 3 nucleotide bases, and they form different instructions, which then correspond to different proteins, which then correspond to different amino acids that code for proteins. So lastly, once all of these codons in a big long strand that form the mRNA, or the premature mRNA, a terminator, which is another spot, kind of like the promoter. Basically what it is it's a spot that signals for the RNA polymerase to disattach, and then a strand of pre-mRNA is left. And after that, the mRNA is still not very useful because there's things inside of it called introns and exons that were copied from the DNA. Introns are bits of DNA that were copied that they're no use to the coding for proteins, whereas exons are the useful parts that you want to keep. So basically what happens is the introns are spliced out, and the introns are cut out and the exons are spliced together, and then a cap and a poly A tail, or a methylated cap and a poly A tail, are then added on, and that's the completed mRNA. Now after that, the mRNA is able to leave out of the nucleus, and it goes into the cytoplasm, where rRNA has formed a ribosome. And basically an rRNA, or the ribosome, reads the mRNA, and it gets corresponding tRNAs with anticodons, which basically are the inverse of the codon that's contained on the mRNA. The tRNA brings amino acids over to the ribosome, and the start codon, which is MET or AUG, basically that starts signals for the process of translation to start. And so the mRNA move through the ribosome. The first codon goes into the P site on the RNA, and the next codon is in the A site. And basically what happens is tRNA match up with their mRNA counterparts, and that puts the correct amino acids in a sequence up at the top of the ribosome. And once the amino acids in place, the mRNA is free to move along one more, kind of click down, the tRNA leaves, and the A site goes to the P site, and a new tRNA comes in. And basically this process allows for the amino acid to form a big long polypeptide chain, which then turns into the proteins that are required for the organism. So basically this protein this protein forms the blue feathers on the penguin, and so basically that's how the penguin becomes blue. But this blue penguin has a baby with another blue penguin, but they're both carriers of a mutated trait. And now this mutated trait doesn't necessarily affect them, but when they have their baby, basically one of the codons was affected by another nucleotide base went in, so a frame shift mutation happened, and what that caused is now the new baby penguin is pink. And basically that's what happened. Thank you very much.